Hello, this is a rehearsal of my thesis defense, and I'm a PhD candidate in the Western University in the Department of Pathology, and I'm going to give a presentation on my thesis, which entitled Development of Non-covalent Functionalization of Carbon Nanotubes for siRNA Delivery. Here is the outline of the presentation today. First, I'm going to give a brief introduction to siRNA and carbon nanotubes, or CNT. Next, I'm going to show you three different approaches on using the functionalized nanotubes for delivering siRNA. First one is lipopolymer, DSPE, PEG, PEI modified nanotubes for in vitro and in vivo siRNA delivery. The next one is PEI succinic anhydride modified nanotubes for topical siRNA delivery in melanoma. The last one is folic acid bearing PEI modified nanotubes for target siRNA delivery into tumor. So let's talk about siRNA. siRNA is an oligonucleotide, which it has around 21 nucleotides or base pairs. It could induce post-transcriptional gene silencing, and it leads to mRNA degradation. The advantage of using siRNA is its specificity to target mRNA. It can be used to take out the action of a pathological protein, say a protein responsible for growth and proliferation in cancer. So we can stop or inhibit the cancer from growing. Also, it is easy for chemical modification. However, the process of transfection, which is to put siRNA into cells, is not easy. Why is that? First of all, because of its nature, it's an oligonucleotide, which can be degraded by the RNAs which exist almost everywhere in our body. So it loses its function before it gains entry into the cells and carries out its function if it is not protected. Second, even if it is not degraded before it reaches the cells, the spontaneous entry of siRNA into cells is unlikely due to the polyclinic nature of siRNA. The cell membrane is also negatively charged, and so they tend to repel to each other. Thirdly, the size of siRNA is big. As I mentioned earlier, it is around 21 base pair, and which is around 15,000 Dalton. Diffusion of siRNA into the cell is not likely. Lastly, depends on how we administrate the siRNA. The localization of siRNA in the pathological tissue is not easy because it can diffuse to everywhere, especially for this systematic administration. So let's talk about how cells take up stuff. Cells do not take up random stuff floating around easily. Scientists have discovered and developed various methods to lure the cells to take up stuff. In this presentation, I focus on the nano-sized material uptake. Generally, you can divide it into two categories: targeted uptake and non-specific uptake. Targeted uptake, like the receptor-mediated endocytosis and phagocytosis. There's only a certain range of sizes of substance can be taken up for both. Also, different cells exhibit different receptors and endocytotic pathway, such as phagocytes, like macrophages, they exhibit phagocytosis, but not the other cells, like cancer cells or other kind of cells. Also, different cells express different receptors, and it can be exploited for targeted delivery. Non-specific uptake of positively charged particle and particle with lipophilic ligand are usually the way for the nano-sized material to be taken up by the cells. This method exploits the physiochemical property of the cell membrane instead of the conventional biological pathway. Currently, there are some transfection methods available for nucleic acid delivery or siRNA delivery. The most important vector, or the viral vector, has the highest transfection efficacy, but it may induce lethal side effects such as inflammatory response and extensional metagenesis. There are also some physical methods such as using the siRNA, electroporation, and sonoporation to deliver siRNA, but the transfection efficacy are low. Hydrodynamic injection is a notable method, but it is mostly restricted to rodents because injecting the same amount of fluid as the blood could be very dangerous. In our lab, we do hydrodynamic injections to mice, and sometimes the unlucky mice died in a couple of minutes after the injection. We can use animal mice, but it's not a practical method for introducing drug for human. Lastly, we can use some chemicals such as lipids, polymers, or other nanomaterials for delivering siRNA. Depending on the materials, the transfection efficacy and toxicity varies. So, the delivery of nucleic acid, in brief, it seems like it is not that easy. So, is there a solution? So we give up, or maybe we can try a new method. There is a new material for a new method. Carbon nanotube is a relatively new material, which is a carbon electrode. It was discovered by Iijima in 1991. Some of the other carbon electrodes are graphite, the best friend of girl diamond, amorphous carbon, fluorines, and graphene. Let's take a closer look on the carbon nanotubes. In general, there are two kinds of carbon nanotubes: single-walled carbon nanotubes, as shown on the left, and multiple-walled carbon nanotubes on the right. Here is a short list of the properties of carbon nanotubes. As mentioned in last slide, they can be single-walled or multiple-walled. Second, they have a high surface area and aspect ratio. The next one is they do not dissolve well in any solvent, but you can disperse it in a solvent with the help of a polymer such as DNA, which can make it solubilize in water. Finally, they can be functionalized by covalent method, such as adding some functional group onto the tube. They can also be functionalized by non-covalent method, such as by DNA. So, can we use CNTs to deliver nucleic acid? Very interestingly, yes. In 2004, patent holder used CNTs for delivering plasmid DNA encoding GFP, and surprisingly, some of the cells exhibit green fluorescence. Later on, researchers found out CNTs can get into the cells independent of cell type in a similar degree, which is diffusion-like. It's another surprise because it's known that quite a lot of cells are difficult to be transfected. And another notable thing is it has a controversial toxicity, basically because it depends on how the CNTs were functionalized. So let's take a look on the cellular uptake of nanotube by the cells. Interestingly, researchers found out the cellular uptake of CNT by TEM observation. The CNTs are orthogonal to the cells. So, in conjunction with the diffusion-like cellular uptake, researchers believe that they gain into the cells like a nano-needle, or someone called it nanopenetration. Later on, researchers tried different methods to modify CNTs and various mechanisms they have found out. First, if the CNTs they were bundled together, it tend to gain the cells by phagocytosis, which is exhibited by a limited type of cells. Also, if the CNTs is longer than 1 micron, even if it is singly dispersed, it will tend to gain the cells by phagocytosis. If the CNTs they are singly dispersed and shorter than 1 micron, it might gain the cells by diffusion-like mechanism. But if the CNTs were conjugate or bind or dispersed by a supramolecular complexes such as protein, then it might gain the cells by receptor-mediated endocytosis. So, what are the technical barriers on using CNTs for nucleic acid delivery, such as siRNA delivery? The most important thing is it doesn't dissolve well in solvent, and you cannot put it into a biological system easily. Also, the insoluble nanotubes are believed to induce the toxicity. It comes from the method of covalent and non-covalent functionalization, or how the nanotubes were functionalized. Also, nanotubes doesn't bind to nucleic acids spontaneously because it doesn't bear any charges. Even the naturally occurring carboxylic acid on the nanotubes, if you functionalize or purify it with an acid, for example, the charge density is very low. So they do not interact with the nucleic acid effectively, and therefore the nucleic acid loading capacity is very low. One of the representative siRNA delivery with non-covalently functionalized nanotube were reported in 2007. New used a lipopolymer called DSPE-PEG, which is covalently attached to siRNA but non-covalently attached to the nanotube. They found significant gene silencing in the T cell, which is very difficult to transfect cells. So what are the advantages of this non-covalent functionalization? It is easy and effective, but the complexes are negatively charged and the uptake of cells are not as easy. Also, the siRNA has to be chemically modified, which is more expensive and is not a universal method for carrying nucleic acid or any kind of siRNA. So how can we modify the design? We can use a cationic polymer or lipid, which is something you can obtain easily. And also, we can choose a polymer which has a known function or toxicity to cells. It can form a positively charged complexes with siRNA, and so more complexes can gain to cells because of the overall charge of the complexes. Finally, the polymers can be modified for achieving our design function, such as targeting some cells that we want. Here is my objective of my study. My objective is to make a nanotube-based siRNA delivery reactor, which has high solubility in water, and it can protect the siRNA and release the siRNA in the cells. It has high serum stability and go to the pathological tissue, and finally, it has a low toxicity. In my project, I used a single wall carbon nanotube for having the enhanced delivery. I choose to use polyethylenimine or PEI, which is a good polymer for nucleic acid condensation by electrostatic interaction. Also, it has high water solubility and it could induce endosomal escape. I also use DSPE-PEG as well as PEG because it's known that it could disperse CNT as well as it can be chemically modified. So we have two different approaches. One is to use DSPE-PEG conjugated PEI to disperse nanotubes, and another one is to use PEI itself to disperse nanotubes. Here is my first study, which is using DSP-EPGPI to functionalize nanotube non-covalently for siRNA delivery. So bear in mind, I'm going to talk about this something like this. This is how I synthesize the DSP-EPGPI or DGI. So basically, it is a very simple amine and EDC activated carboxylic acid reaction. So I made 3 different DSP-EPG to PI ratios. They are 5, 9, and 18. Here is the characterization of the DGI and DGI with nanotube or DGIC. Here is the TEM. It shows like how there is nanotube and they are singly dispersed. And there is also the gel shift assay. It shows how good the nanotubes or DGIC is condensing the siRNA. This one is the state potential, which is one of the criteria to tell the colloidal stability of the DGIC and siRNA complexes. And the higher magnitude it is, the more stable it is. This slide shows the DGIC in delivering siRNA to B16 cells. So you can see that there is a significant gene silencing for all of the DGIC complexes, or the DGIC with the siRNA complexes. Here, DGIC 9 is the best among all of the 3 in terms of gene silencing. The other figure shows the cytotoxicity of the DGIC. Basically, they are all about the same. So we use DGIC 9 for in vivo systematic delivery of with SciFi labeled siRNA, just based because of the in vitro experiments. So the siRNA is tagged with a red fluorescence dye, and we can see the intensity of red fluorescence. So the higher it is, the more siRNA localized in the organ. So most of the siRNA went to the liver, spleen, and some of it in the kidney. Also, there is a little bit in the lung. So this slide shows the gene silencing in 5 vital organs. We can see that there is significant gene silencing in liver, but no gene silencing in other organs. So here is the summary of this project. We developed a new way to functionalize the nanotubes for siRNA delivery. It induced gene silencing in vitro and in vivo for liver. So this one is another project which I use topical method delivering siRNA using nanotubes in the melanoma, which is published in the Biomaterials. Topical siRNA delivery against the skin is difficult because the skin was designed to prevent the entry of harmful substances. siRNA is certainly one of the foreign substances that our skin would exclude. The challenge of transdermal siRNA delivery is mostly due to the hydrophilicity and the lipophilicity balance, and also the stability of the siRNA is a problem because of the active RNAs on our skin. Not to mention like the surface charge or the size of the siRNA, which make it more difficult for delivery into, say, the skin or other target tissue. So in 2010, the year I admitted to Western, scientists used nanotubes for enhanced transdermal drug delivery They use INT and doxorubicin and functionalize the nanotube with PEG or PEG to deliver these two drugs against the skin. Although I didn't with that paper at the time, but yeah, anyways. So we something like this, using the PI for dispersing the nanotubes. But well, PI itself is not very good in delivering siRNA. In 2008, Suchenko modified PI for siRNA delivery, and it's way better than the unmodified one. Therefore, we settled to use the same modification, which is using a succinic anhydride modified PI for dispersing the nanotube. Here is some facts about melanoma. BRAF is usually overexpressed in melanoma, and it is responsible for cell proliferation. In clinical trial, its inhibitor has been used and some success was achieved. So back to our nanotubes. Here is the characterization of the polymer and the nanotubes. So there is succinic acid in the PEI, although you may not able to understand why it is so. And the TEM is better probably because you can see like the nanotubes, they are singly dispersed. And it condenses the siRNA cause of gel swift assay, and you can see the cell potential of the IS, what we call PI succinic anhydride nanotube, ISC. Well, they are relatively stable. And I used the nanotube to down-regulate the BRAF gene on B16 cells, which is mouse melanoma. And also, I compared the cytotoxicity of the nanotube and PI. Basically, they are the ISC is a little bit lower than the PI. So we applied the nanotube with DMSO and Geisero to deliver sci-fi labeled siRNA topically to shave the mouse skin. And in here, it showed decent penetration of the siRNA into the skin compared to the untreated one and the siRNA only and other controls. And it reduced the gene expression on the skin around like 50%. Next, we tested this topical siRNA delivery system on the tumor of melanoma-bearing mice, and the penetration of siRNA is less obvious, but we can still observe it. We analyzed the RNA or mRNA by PCR, quantitative PCR, and the protein by Western blot. And here is a significant downregulation of the BRAF in the tumor, the BRAF gene, and the BRAF protein. So we wanted to see if we can use this topical siRNA delivery system to reduce the tumor progression, and we tested in mouse model. We inoculate the tumor to the mice transdermically at day 0, and then we apply the nanotube with BRAF siRNA, nanotube and BRAF siRNA topically from day 3. And then until around day 25, we sacrifice the mice. So in here, we can see the in vivo cancer progression of the ISC treated melanoma bearing mice. As you can see in here, This one is the control, this one is the untreated mice, and this one is the BRAF nanotube treated mice. And so starting from day 21, there is significant difference of the SI-BRAF treated mice compared to the control. And also at day 25, 25th, we take out the tumor and raise the tumor, and there is a significant differences between the BRAF siRNA treated mice compared to the others. So here is a short summary on this project. We developed a novel non-covalent functionalization of nanotube for siRNA delivery. It could induce in vitro gene silencing and in vivo gene silencing topically on the skin. Also, it could reduce tumor progression in the melanoma model. So this one is my last project. Because of the results from the previous project, we modified the polymer for target siRNA delivery. Previously, we used succinate anhydride modified PEI, and then we further modified the polymer with PEG and folic acid. So again, we are delivering the gene or siRNA in cancer, and our model was again melanoma. So one of the facts we know about melanoma is mTOR, this gene is usually overexpressed. And so we want to target the mTOR using mTOR siRNA systematically. So we conjugate PEG to the PI for reducing toxicity in vivo, and then we attach the folic acid to the polymer because folic acid receptors are usually overexpressed in cancer cells. And we compared also the FA conjugate folic acid or FA conjugate polymer versus non-FA conjugate polymer. So this one is a slide showing how I synthesize the folic acid modified polymer. It is pretty handy. In short, I modified the PEG and into something so I can conjugate the folic acid onto it. And then I conjugate the PEI, modify the PEG, and then conjugate the PEI onto the PEG FA, and then I modify the PEI with succinate acid. Done. So this one is easier because there is no folic acid, and I just react the PEG with some these reactions, and then with PEI, and at the end modified it with succinate anhydride. So here is how the nanotube looks like. It's again the TEM and the gel swift assay and the cell potential. Here is the cytotoxicity of the FGISC and GISC in the B16 cells. Both FGISC and GISC have a lower cytotoxicity compared to the ISC. So this one is slides showing we used FGISC and GISC. We injected this with the side-field labeled siRNA to a tumor-bearing mice. We can see that there is more siRNA localized in the tumor for FGISC compared to GISC. So again, we treat the mice with mTOR siRNA but this time we use subcutaneous injection at day 0, and then starting from day 3, we inject the mice with the mTOR siRNA and the nanotubes once a week by tailoring, and then we test, and then we see the tumor progression. So here we can see the tumor size and tumor rate. On the left, it is the tumor size of the mice, both FGISC and GISC with mTOR siRNA treated mice has a significant reduction in the tumor volume compared to the controls. The same thing, the weight is reduced or inhibited as observed on the day of mouse sacrifice. So here is the Western blot of the tumor tissue from the treated mice. We found a significant reduction in mTOR expression for the FGIS group. Here is the quantification by the band absorption, which is around 60% of downregulation. So here is a short summary of this project. We prepared 2 nanotubes for in vivo siRNA delivery, which is able to deliver siRNA in vivo to inhibit the tumor progression with mTOR siRNA. So, in conclusion, these projects provide carbon nanotubes functionalized by non-covalent polymer association for the purpose of siRNA delivery. In vitro, in vivo for topical and systematic administration of nanotubes with siRNA was demonstrated. For the tested nanotubes with the siRNA, a significant inhibition of tumor progression was found. So this is the end of my presentation. Thank you very much, and this is the people I want to thank, and especially my supervisor. And in this year, I was supported by the CAS program, which is a very great cancer trainee program funded by Professor James Korpanik. So thank you very much. If you have any questions, please feel free to leave a message. Have a good one.